Bismillahirrahmanirrahim. 5090 O-Level Biology, June 12, paper 1-2. And the first 22 questions. This is the first video. Question 1. Some structures found in cells are listed: cell wall, cell membrane, chloroplast, cytoplasm, nucleus. Which structures are found in both animal and plant cells? Well, that's always the first question. Is a very easy question, so as to make you all a little comfortable. So you know cell wall is only present in plant cells, cell membrane is present in plant and animal cells. So both of them, chloroplast is present in some plant cells, not all plant cells. Cytoplasm is present in plant and animals. Nucleus is present in plant and animals. So it was 2, 4, and 5. Cell membrane, cytoplasm, and nucleus, which is present in both plant and animal cells. Then coming on to now question 2. Which process can only occur through a membrane? Yes, for active transport, you need a membrane because if a membrane is present, then there will be a channel protein in it which allows the active transport because it has an ATP binding site because it requires energy. So the ATP is converted to ADP, which is an energy currency. And so you need a channel protein and you need an ATP binding site where the ATP binds and active transport takes place from a low concentration to a high concentration inside the cell, or other way around, could be from inside the cell to outside if it is a low here and high outside, and that will be also active transport. So it can be in either direction, active transport. Then diffusion is, of course, doesn't need a membrane, can happen. Membrane can be or cannot be present, and it doesn't really matter. And osmosis also has to have a membrane. So only diffusion where there was no membrane, and active transport and osmosis. So diffusion means that it's because it said which process can only occur through a membrane. So diffusion, like you take a beaker of water and you add maybe green ink to it, and the ink spreads all around. This is diffusion, and there's no sort of membrane present here in a beaker of water. So this is what we had to understand in question 2. This is a difficult question because identical pieces of potato are placed in sucrose solutions. So we've got, say, 5 sucrose solutions: 1%, 2%, 3%, and 4. And we different concentration, maybe even 5 solutions we have. And after, what we do is we place pieces of potato in it. So we put pieces of potato in these sucrose solutions, and after 3 hours, what do? The mass of each potato piece is measured. So which graph shows the results of this experiment? Now, as I've told you, in every graph, what you do first thing is you add here. So you add 1, 2, 3, 4, and 5. So the concentration in arbitrary units, say percentages, the sucrose concentration is increasing. The mass of potato after 3 hours. Now, in a dilute solution, it will have increased. In the same solution, it will remain the same. But in a highly concentrated solution, it will lose water, so the mass will decrease. Because you have to understand is, let me explain this to you. If you have, this is the piece of potato, and inside the piece of potato is 22%. I mean, the cytoplasm or the cell sap is 2%. So if you have a 1% solution outside, what is going to happen? What is going to move in? And this is going to increase in mass. So a 1% solution will have a bigger mass, and then you put it in a 2% solution. Well, the mass will probably remain the same. So the mass in arbitrary units. So the mass, say, was, we can add something here: 1, 2, 3, 4, 5, 6. The mass, say, was 6 grams. So then the mass has to decrease. Why? Because in the same percentage, and 2% will not have anything. But then when you place it in a 3% solution, what is going to happen? What is going to move out? So it is going to decrease in mass. So the mass is going to slowly decrease. Because if you put it now in a 4% solution, even more water will be lost. So that is why the answer to this was D. You had to understand is mass of potato after 3 hours. So the mass of potato, say in the beginning, and the beginning was whatever the concentration was, that is 1%, 2%, 3%, 4%, 5%, and 6%. So in 1%, it is a dilute solution. So say it was 10 grams. But as you put it in a 2% and then in a 3%, then as it is, you put it in a more concentration solution, it has to lose weight. So the mass of potato should decrease. Now this mass was less. If I put here 1 and I put here 2 and then I put here 3 grams and then 4 and then 5 and then 6 grams. So it was probably 6 grams and then it has become 2 grams. So that is why the answer to this was D. So please always put figures on the graph, and this is the way that you handle the graphs. Number 4, starch is digested to maltose by the enzyme amylase according to this lock, and which is the key. So the key has to be, because we're talking of the enzyme, so the enzyme has to be the lock. So the enzyme has to be the lock, amylase. And starch is the substrate, has to be the key. So this is amylase. So this is lock. The lock is amylase, the ASE, the enzyme. And what fits into it is the substrate, and in this case it is starch, which fits into it and then breaks up into maltose. And maltose would be the product. Maltose would be the product. And question 5, if a condition is a limiting factor for photosynthesis, what does this mean? It means that changing the condition may either increase or decrease the rate of photosynthesis. Yeah, because if you increase the light intensity, it increases the rate of photosynthesis and then it levels out. If you increase the temperature to above the optimum, it will denature, the rate will decrease. But if you increase the temperature from a low temperature till the optimum, there's an increase in the rate of photosynthesis. So you have to think of all the graphs. This is the temperature graph. Temperature in the graph, what happens to the rate of photosynthesis? Initially the rate increases and then it decreases. Then if you look at the light intensity graph, the rate increases and then levels out. So limiting factors would be light intensity in this situation here, till here to here is the limiting factor. So A to B, light intensity is the limiting factor, whatever is on the x-axis. And then it becomes constant, and then some other factor is limiting, like temperature or carbon dioxide concentration. So limiting factor is when it changes in the condition may either increase or decrease. And then, of course, this was totally wrong. Changing the condition will not affect the rate of photosynthesis. This was changing that will only decrease. So this was wrong. Increase, this was wrong because it can increase or decrease the rate of photosynthesis. Temperature increasing does decrease the rate of photosynthesis. Question 6. The diagram shows cells in a section through a leaf of a green plant. No cell contents are shown. Which cells contain chloroplast? Done this in a previous paper as well. It is 2 and 4. So both the palisade mesophyll also contains, spongy mesophyll also contains, and guard cells also contain. So there are three. So what does it not contain? Cuticle does not have any chloroplast. Upper epidermis does not have any chloroplast. Lower epidermis does not have any chloroplast. Only the guard cells have chloroplasts. In question 7, now it is very obvious something was missing. Two aquarium tanks are set up as shown. Now you have to realize air bubbles were present in both. And air is providing the oxygen, so you're not going to be worrying about the oxygen. So oxygen was totally wrong. Because in both situations, air is being present. Then it says there's a pump and there's snail and there is some plants and there's snails. And the second in tank X contains animals and plants. Tank Y contains only animals, no plants. After a week, all the animals in tank Y show signs of distress. This is because the animals have run out. If the animals are distressed, that means there's something which is they are not getting, which they should get. And basically, the two things which they need is food and oxygen. So oxygen is present because there's air being bubbled into it. So the food must have finished. So very obvious question, but difficult, I guess, for some students. Then it says which process is stimulated by adrenaline in the cells of the liver? Breakdown of glycogen, increasing the blood glucose. Adrenaline is a anxiety hormone. It is produced in stress. So what does it do? It makes the liver convert the stored glycogen to glucose, and the glucose level increases, and that is how you can able maybe in a fight in an action movie when you have to do a lot of muscular action, so you will need more glucose in your blood. Then question number 9, which facts about proteins makes them essential components of the human diet? They provide amino acids for cell growth. So proteins are not an energy source. It's carbohydrates which are an energy source, and fats are also an energy source. They're used in synthesizing hormones. Sorry, hormones are needed, yes, but there's some hormones which are not made up of proteins. They can be stored as a food reserve. No, proteins cannot be stored at all. Carbohydrates can be stored as glycogen or as fat, but proteins cannot be stored at all. It says which fact about proteins makes them essential component. Essential means you need it in a large, for a large important function for the body to remain normal. So that was, they provide amino acids for cell growth. Cells grow and then divide by mitosis. So cell growth, it said, and that was important. Number 10, transpiration increases when conditions become what? Less humid. Why? Because if outside there is less humidity, if there is 1% humidity, and inside there is 3% humidity, then of course the water vapor is not going to diffuse about. So less humid, so increases. Now in this situation, of course, this will move out because inside is 3% and outside is less humid. So less humid outside and more windy. Why windy would help? Because it will carry away the water vapors which have been diffused out. And these water vapors will be carried away, so a continuous gradient will remain. The inside air spaces have 3%, outside it has 1%. So the water vapors are going to diffuse out, and because there's wind blowing, so it's going to carry away the water vapors. So there's less water vapors outside the leaf. So less humid and more windy. The other choices were more humid was wrong. Why more humid was wrong? Because you see, if inside the leaf we have, now it becomes very simple. If you look at it, inside the leaf you have 3%, and outside the leaf you have 4% humidity. Well, water vapors are not going to diffuse out. They always go down the concentration gradient. So no water vapors are going to diffuse out. But naturally, if we have inside 3% and outside 1%, then water vapors are going to diffuse out. And more windy because it's going to carry away the water vapors. 11, the diagram shows a section through a green leaf. The arrows represent the movement of. Now you can see what is happening. These things are coming out of, this is the xylem. You can see these thick walls. And this is the xylem. So the water is coming out. So water during transpiration. Why? Because it's also coming out. And it couldn't have been other thing. Carbon dioxide during respiration. Well, carbon dioxide will respire and it will of course be used in by the plant for photosynthesis. If it is broad daylight, then carbon dioxide is produced during respiration, but then it won't enter the palisade cells. It's some of it, but then it won't come out of the leaf. Oxygen during photosynthesis, yes, but oxygen doesn't come out of the xylem vessel. This is coming out of the xylem vessel. This is the xylem, all this is the xylem. Then sugars during translocation, well, that's again in the phloem, and sugars don't move out of the leaf. So you have to think of two things could move out of the leaves, either water vapors or oxygen, or some sort of a gas. Question number 12. The diagram shows the direction of blood flow in the human body. Please remember, whenever you see this question, you should do this sort of labeling, which I am doing on the paper. You are allowed to do this. It's rough work on the paper because your papers stay here. It's only your answer sheet which goes back to Cambridge. So always write on it. So left side of the heart to the capillaries and organs of the body was aorta. Then capillaries in the organ to the right side was the vena cava, and then right side to the lungs was the pulmonary artery, and then of course pulmonary artery, this was the pulmonary vein. So which numbered stages have blood containing the most oxygen? Well, it had to be this side, the aorta and the pulmonary vein. So it was 1 and 2. So it was 1 and 2. Question 13. The graph shows pressure changes in the left atrium and in the left ventricle during one heartbeat. Now, what we have to understand is that the graph is the typical pressure graph, and we have to see the point X. What is the state of the valves in the heart at time X? Now, what do we have here? We have the atrium here, and we have this is the atrium pressure, which is a very low pressure circular part of the heart, which is under low pressure, is not very high pressure, because you can say pressure is here, so it's a very low pressure up to this mark. Then we have the ventricle, which is I'm coloring in green, and then you can see how the pressure is rising and the pressure is falling because the ventricle is emptying, and then of course the heart cardiac cycle takes place. Now it says what is the state of the valves at X? It was the answer to this is B. Why is it B? Is because you see these valves are closed and these are open. Now what is happening is if you go to look at the diagram of the heart, this is the left side. So this is the left atrium, and then this is the atrioventricular valve or the bicuspid valve, and this is the left ventricle. Now what we have to understand is that the pressure is rising, and then you see the aortic valve opens, and the left ventricle now, as it is increasing, is because the left ventricle wall is exerting pressure, but then the blood is flowing into the aorta. So as the blood is flowing into the aorta, so this is emptying. That is why the pressure has started to fall. And at this point, what has happened? These valves are closed, but these semilunar valves are open because blood is going into the aorta. And of course, it won't go back into the left atrium. So that is why the atrioventricular valves or the bicuspid valves are closed. Because we don't want, when the ventricle is contracting, we don't want blood to go back into the atria. We don't want it going back into the atria. So this was the answer to it, and this is what you had to figure out. Then coming on to question number 14, what is the difference between plasma and tissue fluid? Plasma is the one which is inside the capillary. And I'm making this dotted line to make you realize that this is a capillary because capillaries have holes in their walls and things can move out of it. But outside this, you have the body cells. And surrounding the body cells is tissue fluid. So all this is called tissue fluid. So it says what is the difference between more protein molecules, fewer protein molecules? The reason why is that protein molecules stay inside. They cannot come out. They're large molecules, so they cannot come out. So this is what you have to understand about it, that it will have more and less protein molecules. Some do leak out, but the very tiny ones. What are the products of aerobic respiration in yeast? Aerobic respiration in yeast, there's no alcohol, there is carbon dioxide, and there's no lactic acid, and there's water. So it's a normal process of aerobic respiration. Is glucose plus oxygen gives you carbon dioxide plus water plus energy. So it is the normal process of aerobic respiration. Coming on to 16, the diagram represents the human respiratory system. Which structures contain muscles that contract when breathing? So Z was the diaphragm muscle and Y was the intercostal muscle. And this is, of course, what you have to understand is it's a very simple way that you understand it. These are the outside ones are the external intercostal muscles, and these are the internal are this side here, which you can see here. So the fact that you see this internal and this internal, so it's very easy because you see you've got to realize is that they have to show you the ribs. The ribs are these, and then the two structures, one on either side, will of course be the external and internal intercostal muscles. And these of course are the ribs, which you see as one solid sort of when you cut the rib open. This is what it's going to look like. Then question 17, which substance builds up in a muscle as a result of anaerobic respiration? Lactic acid. Lactic acid builds up as a result of anaerobic respiration. 18, the diagram shows apparatus used to investigate breathing. Now M is the one which goes into your mouth, and at the start both tubes contain orange hydrogen carbonate indicator solution. The solution is orange when atmospheric air is passed through it. The solution changes to red when air with less carbon dioxide passes through it, and the solution changes to yellow when air with more carbon dioxide passes through it. What changes occur to the hydrogen carbonate indicator solution in tubes X and Y when a person breathes in and out? Now, first of all, you've got to understand is that the outside air enters through this tube, goes through this chemical, and then it goes into your mouth. And you see these rubber tubings here. And these rubber tubings, actually, you close this one when you're breathing, taking this process, when you're inhaling in. And then when you are exhaling, what do? You close this one, and then the air that you exhale is going to enter here, and then it's going to enter here, this tube. So this tube goes and drifts, and then it's going to come out here. So tube X has normal. So when tube X is normal, so its solution is orange when atmospheric air passes through it. And when more carbon dioxide, so this would be orange. But when more carbon dioxide, it says the solution changes to yellow in area with more carbon dioxide. This one is going to become yellow. So that was the answer, orange and yellow, and the answer was D. Then coming on to question number 19. The diagram shows a pair of chromosomes from the same cell. What do the lines P point to? So P is the site of a gene made up of two or more alleles which might be different. Could be the same. There could be big A on it. There could be small a on it. There could be big A, small a on it. So all three possibilities are present. Because it says gene, we don't know how many alleles it has, but if it has two alleles, the gene is made up of two or more alleles. Like for blood group, we have three alleles, IA, IB, and IO. So this is how we have to do these questions. Next question. Can be removed from the blood by kidney machines. Urea and some salts only. The rest, of course, cannot be removed. Glucose, impossible. We don't need to actually remove glucose. Glucose, glycogen, glucose, protein. Again, here is one large molecule glycogen, large molecule protein, urea, vese can be removed from the blood by kidney machines. So urea, yes, but then glycogen cannot be removed. So the reason of these and these are all wrong. And the only answer was urea and some salts. Then coming on to question number 21. The diagram shows skin temperature of human when exposed to warm air and then exposed to cold air. Naturally, you enter this room is very hot, so your whole body gets heated up because of the environment temperature. But if you're in cold air, you see your extremities get cold. All there's vasoconstriction of your distant body parts, and your core body, which is your heart and your lungs and your liver, this will remain sort of, it will be kept at a higher body temperature. So now it says above 26 degrees was the one which is the shaded area, which in cold area was this shaded area. So this is the core body. And then in warm air, the whole body was 20 was 26. No, it was about 26. Here the other one, this one, the one which was white was 26 degrees or below 26. So this was the one which was in white, and this is the one which we were looking at cold air. What causes the observed changes in skin temperature on exposure to cold air? Less blood flowing just below the skin. So what causes the observed changes in skin temperature? How did the skin temperature become sort of 26 or below? Is because less blood flowing. So vasoconstriction. So this was questioning whether you understood vasoconstriction or not. So question 22. A child is frightened by a loud noise and shouts to help. In which order are the different types of neurons involved in this? So first is the loud noise. So he's got to be hearing it, sensory neuron. And then it's going to be a relay neuron. And then it's going to be a motor neuron and shouts for help. So he uses the muscles, larynx, and the muscles to shout. Shouting is an active process. So motor neuron is involved. Motor neurons supply muscles and glands. So shouts for help. So he needs to do some sort of muscular activity to shout for help. You've got a throat and you've got larynx and you've got to scream out. So the air has to be forced out. So that finishes this paper. And this is the last paper that I'm doing before the exams. If there are any queries, please let me know. You all have my number and you can access. I'm going to give you my number at the end of this video. So please everybody revise the second, do the second video as well after this, and then this will be the last paper that I'm posting. on my YouTube channel. And thank you very much.